Hello everyone, this is part 4, chapter 8, that is unit cell, the unit of life. This is unit 3rd. In this part we have to discuss vacuoles, characteristics, then we have to see in detail, that is detailed structure of mitochondria and its functions. Then we will go for plastids and especially the chloroplast, its detailed structure and function we have to discuss. This is part 4, chapter 5. Unit 3, name of the chapter is, this is chapter 8, this is cell, the unit of life, and this is part 4. Part 4 of chapter 8 will include discussion regarding vacuoles. Then we have to see structure and function of mitochondria, and in the last we have to see structure and function of chloroplast. Vacuole, I told you in the beginning of this chapter part 3, that this is a part of endomembrane system. Endomembrane system, you know, yes, this is membrane system. These are, that is, interrelated, and what we have in endomembrane system you must be knowing now. That is, one is endoplasmic reticulum, one is that is Golgi body, one is that is lysosome, and one is that is vacuole. Okay, we have to see detail of vacuole now. Vacuole is membrane bounded space found in cytoplasm. It contains, if we see, that is, vacuoles are, in case of plant, the vacuoles is large and this is centrally placed. Because of this, the cytoplasm and the nucleus will be shifted towards periphery, you know it. It contains water, sap, excretory product and other material which are not useful to the cell. The vacuole is bounded by single membrane and this membrane is known as tonoplast. In plant cell, the vacuole can occupy about 90% of the total volume. You know that this is the cell here, that is this is the vacuole we have inside, and here the cytoplasm and the nucleus will be shifted. A large central vacuole is found. In case of plant, the tonoplast, it facilitates transport of number of ions, other material against concentration gradient into the vacuole, because in plant cell we do not have, that is, contractile vacuole. The substance will be collected here. Hence the concentration is significantly higher in case of vacuole than in cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion. In many cells, as in protist and food, vacuoles are also formed, that is, which are formed by, that is, when food will get fused with lysosome, that will be formed, that is food vacuole. Is it? Come to the next part, very important one, that is very interesting one, mitochondria. You all know, in earlier classes you have seen, mitochondria is called, that is, powerhouse of cell, because the energy will be generated here, which is required by cell. Mitochondria, also known as chondriosome or sarcosome. Mitochondria were discovered by Kolliker in striated muscles of insect. You have to remember it. Present name was given by Benda. Benda called these as, that is, mitochondria. Altman called these as bioplasm. Sometimes the question related to this text also come in examination. Very, very important one. Mitochondria are stained with Janus green stain, and this stain is known as vital stain. If we want to see mitochondria and want to study its function, then we have to keep in live form, and the live stain is Janus green 1. These are absent in prokaryotes. You know, membrane bounded organelles are absent. Mitochondria are secondarily lost in case of RBC of the that is mammals. RBC initially were having this mitochondria, but now they do not have mitochondria and they are used to respire anaerobically. Mitochondria, the single is mitochondrion. Unless specifically stained, these are not easily visible under microscope. That is, staining is required. Number of the mitochondria per cell, it is variable. Definitely, the cell which is physiologically more active, which requires more energy, will have more number of mitochondria. In terms of shape, size, considerable degree of variation is there. Typically, if we see a typical mitochondria, it is just short-shaped or cylindrical, having a diameter of 0.2 to 1 micro, and the length is about 1 to 4.1 micro, that is meter. Is it right? This is diameter, that is length. Now you see ultrastructure, interesting one. If you see Let this is mitochondria 1. Mitochondria is double membranous one. This one is outer membrane one, and this is the inner membrane. Inner membrane, it invaginates inside the matrix or inside the mitochondria like this, and this will be invaginated like this. Is it? That is inner membrane. That will make surface area, increase surface area will be there because of that is inner membrane. Is it right? Inner membrane this is. So this is a double membranous structure, that is outer membrane and the inner membrane, that divide the lumen distinctly in two aqueous compartment. This is the outer one and this is the inner one. This is the outer compartment one and this is the inner compartment. This outer compartment, it is filled with dense homogeneous substance, and it is called matrix. Here we have to see the site where Krebs cycle will take place and where the glycolysis will take place. So here we see in this diagram, I'll tell you here, this is outer membrane, this is inner membrane, this is the space in between the two, that is known as peri-mitochondrial space. This is peri-mitochondrial space. This is inner membrane, and in inner membrane, it is invaginated inside this, that is known as matrix. It is called matrix. You see that this invagination here, these invagination finger-like structures, these are known as cristae. Cristae will increase the surface area of inner membrane. On cristae you have to see the particles are there. You have to see detail of this. That is, these particles are known as F1 particle or also known as oxyosome. Detail of this I'll tell you little later. But you have to remember one thing here which is more important, that here in cytoplasm, that is the glycolysis, if you see, that is function of mitochondria. Glycolysis will take place in cytoplasm, and then this pyruvic acid will enter into the mitochondria, and the site of Krebs cycle will be this one. That is Krebs cycle, TCA cycle, or Krebs cycle, that will take place there in mitochondrial matrix. So you have to remember in time to come, detail of this we have to associate with this. You see the same thing we have here. Come on. That is the outer membrane. It forms a continuous limiting boundary of the organelles. The inner membrane, that is number of enfoldings are there. Enfoldings are known as cristae. The cylinder will be crista towards the matrix. Is it? The cristae increases the surface area, as I told you. The cristae and the inner face of the membrane, that is studded with numerous spherical knob-like protuberance called elementary particles, also called as particles of Fernandez and Morin, or also known as F1 particle, or also called as oxyosome, also known as oxyosome. Is it right? It is also called oxyosome. Okay, oxyosome this is. See what we mean by here. You have to see. This is inner membrane. Okay, and on inner membrane, these are the particles which are found attached to this inner membrane. These are inner membrane, and these are the particles attached to this. The name of these particles, I can repeat once more: elementary particle, that is particles of Fernandez and Morin; F1 particle, oxyosome. And this particle, you see, it is having three parts. This is the head, this is the stalk of this particle, and this one is the base of this. We have to see. This F1 particle, basically, that is head piece of this, contains ATPase enzyme, and that is ATP synthetase enzyme, which brings about oxidative phosphorylation. Detail of that you have to study in plant physiology in respiration, but here the ATP will be generated here. In case of this, in F1 particle, ATP will be generated. Okay. The two membranes, they have their own specific enzymes associated with the mitochondrial function. You see, the matrix also possesses single circular, DNA molecule, a few RNA molecule, ribosome 70S type, and the component required for the synthesis of protein. Very, very important point we have here. We have to go little back in here. This is that is mitochondrial matrix. This mitochondrial matrix, it is having its own DNA. It is having that is 70S type of that is ribosome. This DNA that will be a prokaryotic DNA, that is this ribosome is also prokaryotic DNA. This with the help of this DNA then and it is having what? That ribosomal, that is it is having RNA also. All these three together that they will be able to make some of the protein required by this, that is mitochondria. The protein synthesis will take place in mitochondria just like a prokaryotic cell. So here important one is that is mitochondrial matrix is having DNA, 70S ribosome, RNA and different type of enzymes that is required for protein synthesis, which are that is urgently required by mitochondria. You see here, this I told you here, very important point this one is. And origin, that is mitochondria, these are self-duplicating one, just like bacteria, as the bacteria divide by binary fission. In the same way, the mitochondria can divide by binary fission. New mitochondria arise by division of the H61. They cannot originate de novo. Mitochondria are believed to be bacterial endosymbiont. It is believed a time was when this mitochondria was just like a prokaryotic cell, just like a bacteria. In due course of evolution, that it entered inside this system, and here the mitochondria, even now after entry, it has retained some of its prokaryotic character. It is having some autonomy or semi-autonomous. It is dependent on cell, no doubt, but certain protein can be synthesized by mitochondria. It is known as that is endosymbiont. Is it? Important point this is. Now you see, as I told you, they have their own nucleic acid, sulcular double-stranded DNA and RNA, and that one is prokaryotic type, as I told you now, prokaryotic DNA. 70S, this is also found in prokaryotic cell. They show binary fission, just like that is bacteria, and their membrane resembles with that of bacteria. Outer membrane of the mitochondria has porins. Porins, these are the large pores, as are found in case of that is bacteria and chloroplast both. Right? Come to this now, that is semi-autonomous nature. I told you there in detail, because this mitochondria, which was a time was when this mitochondria was just like a bacteria, it was just like a bacterial cell, prokaryotic cell. In due course of time, mitochondria has entered into this system when the cell was to be originated, cell was to be formed. So this mitochondria is found in eukaryotic cell now here, and this mitochondria, as it is having its own DNA, it is having its own that is RNA, it's having own enzyme, it is having its own ribosome. Therefore, this mitochondria can prepare some of the protein. Mitochondria are dependent upon nucleus and cytosol for raw material. Therefore, the structure and working of the mitochondria is partially function controlled by nucleus, but the mitochondria can make some of the proteins. That is, therefore it is known as semi-auto. It's not autonomous. It depends on cytoplasm and nucleus for many of the raw materials, but it can make its own protein. That's why it is known as semi-autonomous one. Right? Functions of the mitochondria, powerhouse of the cell. I told you, that is, this is mitochondria. Inside mitochondria, what will happen? This is the outer membrane and the inner membrane. Pyruvic acid, aerobic respiration, you have to study. Pyruvic acid as a result of that is glycolysis, that is, it will enter into the mitochondria matrix. Here, that is TCA cycle will be there, as a result of which TCA cycle, that is energy rich compound will be formed, which will go to this, that is F1 particle, and where this energy will be released. Detail of this we have to see in physiology respiration chapter. powerhouse of cell energy is generated. They bring about to release this energy. Oxidation of carbohydrate, protein and fats will take place to release energy. This is the site of aerobic respiration, where Krebs cycle will occur in matrix, while the electron transport system and oxidative phosphorylation that will take place on inner membrane, that is in F1, that is particle. Is it right? This is brief, that is structure and function of mitochondria. Now we'll proceed to the next, that is plastids. Large sized, that is cell organelles. Haeckel gave name plastid. Plastids are largest side of type of cell organelles. Plastids are found only in plant cell and also in euglenoids, that is a protist. You know, euglenoid is a protist, that is the chloroplasts are found in this. They are easily observed under microscope. Important question. The cell organelles which can be seen by simple light microscope, that is plastid. They bear some specific pigment, therefore they impart specific color to the plant. Based on type of pigment, the plastids are of three types: chloroplast, chromoplast and leucoplast. Leucoplast, these are, that is colorless. These are found, that is in colorless part of the plant like root. They have major role in storage of the food. These are green in color, and chromoplast, these are, that is colored. Okay. Chloroplast contains the chlorophyll, carotenoid pigment, which are responsible for trapping of the energy, that is chloroplast, that responsible for photosynthesis. Chromoplast, they contain fat soluble carotenoid pigments like carotene, xanthophyll, and others. These are present in chromoplast. They give part of the plant different colors except green, yellow, orange, or that is red color. This is found especially in petals, that is petals are colorful to attract the insect. Chromoplasts are found in, that is petals and other green part of the plant. Okay. Leucoplast, as I told you, these are colorless plastid. They varied in shape and size. They store, that is food stored nutrients. For example, amyloplast stored carbohydrate, amyloplast, that is as in case found in potato. Elioplast, they store, that is the oil and fats, where the aleroplast, they store the protein. So leucoplast, the basic purpose is, that is storage of, that is food, different type of food. That may be amyloplast, that may be, that is elioplast, or that may be, that is aleroplast. Now we have to see detailed structure and function of the chloroplast. Now you see, majority of the chloroplast of the green plants are found in mesophyll cells of leaves. Structure, I told you, anatomy of the leaf I told you, especially in case of lateral dicot leaf, that is, this is the epidermis we know, this is the upper epidermis, that this one is the lower epidermis. Now you know, that is the tissue in between these two epidermis is known as what? It is called, yes, it is called mesophyll. Especially the layers of the mesophyll which are towards the upper epidermis, these are elongated cells, and what we used to call, can you recall? That is known as, that is palisade parenchyma. And this palisade parenchyma, they have, that is huge number of chloroplasts which will help in photosynthesis. So it is found in mesophyll cell of the leaves. The chloroplast, you see, these are lens shaped, oval shaped, spherical, discoid, or even ribbon, depending on the type of organism. The shapes are different. The length may be typical. Chloroplast length is 5 to 10 micrometer. The width is 2 to 4 micrometer. Number, it varies, 1 per cell. For example, that this is the Chlamydomonas we have. It is having only one, that is chloroplast inside, that is as in Chlamydomonas. A green alga, and it may be 20 to 40 per cell of mesophyll. That this is mesophyll cell. So in this one cell, that may be 20 to 40. The shape detail is not required. In algae, I told you different type of chloroplast. As in case of Spirogyra, that is chloroplast is ribbon shaped, and different shapes are found. Like mitochondria, chloroplasts are also double membranous structure. That is, this is the outer membrane and this is the inner membrane. You see the difference? outer membrane and the inner membrane. But the inner membrane is not projected like Christie here. Of the two, the inner chloroplast membrane is relatively less permeable. The outer membrane, it is having the porins like that is mitochondria and bacteria. The space limited by the inner membrane of the chloroplast, very important one, that one is known as that is stroma. What is this? It is called stroma, right? And in stroma, what we have, number of organized flattened membranous sac thylakoid. Children, you see that these are the sac-like structure. These are sac-like structure placed one above the other like this. These are known as thylakoid. These are present in stroma. Thylakoids are arranged in one above the other in stacks, and the pile of, just like pile of coils, and this is called grana. That this is grana. So there may be so many grana, and the grana, these are interconnected by that is stroma lamellai. Is it intergrana thylakoid? There are flat tubules called stroma lamellai. These are stroma lamellai, which are connected thylakoids of different grana. The membrane of the thylakoid that enclose space called lumen. You see, in thylakoid, let this is thylakoid, that is, here it has photosynthetic pigment which will trap the light energy. That's why thylakoid, it has become a place where light reaction of photosynthesis will take place. Detail of this we have to say in, that is photosynthesis, when we will discuss about that is this physiology. So this is the lumen of the that is thylakoid, and this thylakoid membrane, it is having the pigments here. These are the pigments. The stroma of the chloroplast, this is the stroma. It contains that is various type of enzyme required for the synthesis of carbohydrates and protein. We will see detail of this now. You see here that this is outer membrane. Yes, this is inner membrane. These are the grana. Just like coins have been placed one above the other. The top one is known as that is thylakoid. This is thylakoid. If I am going to make that, this is thylakoid 1. This is thylakoid. This is having that is the lumen inside, and here it is made up of, in the same way just like plasma membrane is having that is bimolecular lipid layer, is it? And it is having the protein also, and in between these that is the photosynthetic pigments are there. That these are the photosynthetic pigments in this layer thylakoid. This is stroma lamellai, which will connect the that is two grana, and here that is this one is that is stroma. In stroma we have what we have here. We have that is different enzymes. You know, children, that is stroma is the site of dark reaction where food will be formed. That is, it is the site of dark reaction. You know, photosynthesis has two type of reaction. One is light reaction, one is dark reaction. Site of the light reaction is that one is thylakoid, and the site of dark reaction is that is stroma. So we have to correlate the that is function also. You can see just a brief account I'll give you here. This is the thylakoid. This is the thylakoid. This one is the granum. Okay. Granum, as I told you, it is having the photopigment. It will trap the that is light energy. The water will go as inside. There will be photolysis of water. What is this? That is photolysis of water will take place here. Light energy will be trapped. As a result of it, the power will be formed, and this power will be in the form of ATP and in the form of NADPH2. So this is the part of that is light reaction. In light reaction, that is there is what? Photophosphorylation, formation of ATP, photolysis of water, and the oxygen is released as byproduct. This power is formed. This power will be utilized in dark reaction. That will be dark reaction which will take place there in stroma. So in dark reaction, this power will be used up. ATP and NADPH2 will be used up. Carbon dioxide will be trapped. Carbon dioxide will be converted into food. The sugar are formed. This is the function of that is chloroplast. Is it right? Little part is left. You have to go for this also. That is, it contains double-stranded circular molecule and ribosome. Similar to this mitochondria, this is also that is semi-autonomous organelle because it can multiply at its own. It can that is prepare some of the protein required by this. This is the end of this chapter, part 4. Okay. Thank you.